Hello everyone. Hope you all are well in this pandemic situation. Thank you a lot for all the responses you have shared with us on our previous video. Now, as I said in my last video, today we'll be discussing a briefer details on structures surrounds the cytoplasm of a cell. That is cell membrane and cell wall. Now, to begin with the cell, we all know that every cell, whether it's a prokaryote or an eukaryote, every cell consists of cytoplasm, and outside the cytoplasm they have a membrane which is known as cell membrane, or we call it also as a plasma membrane. Now, many cells other than animal cells such as plant cells, they also have a structure outside the membrane which is known as cell wall. It also acts as to protect the cell mechanically from its environment, and it has an additional layer to protect the membrane. Now today we are going to discuss about this cell membrane and cell wall. Now, to begin with cell membrane, we need to know what is cell membrane and what is their chemical composition. Next we'll come to know the structure of cell membrane and then the functions and the models of cell membrane. Now, what is cell membrane? So the cell membrane or a plasma membrane is a biological membrane separating the interior of a cell from its outside environment. We also called it as plasma membrane or plasma lemma. Now, the term cell membrane was coined by C. Nageli and C. Kramer, whereas the term plasma lemma was coined by Jacques Chloe. We'll come to know what are the chemical composition of cell membrane or plasma membrane. Now, plasma membrane is mainly composed of lipids, and there are four major class of lipids commonly present in plasma membrane. The most abundant one is phospholipid. The others are sphingolipid, glycolipids and sterol. All of them are antipathic. Cholesterol is abundant in mammalian cell, but in prokaryotic cell they are absent. Now coming about proteins, there are mainly two types of proteins: the peripheral or extrinsic proteins and the intrinsic or the integral protein. Now, peripheral protein, they lie just outside and the inner surface of phospholipid bilayer and are loosely attached by weak electrostatic bonds, so can be easily separated by mild treatment of salt solutions. They form about 30% of proteins. For example, ATPase, acetylcholinesterase. And the next is the intrinsic or the integral protein. Now, they are partially or completely embedded in the phospholipid bilayer. They are held by ionic interactions, so that can be separated by drastic methods like solution of detergents, and they are almost like 70% of the membrane proteins. For example, the cytochrome oxidase, rhodopsin, etc. Now, there are many other structural proteins of plasma membrane. They actually form the backbone of membrane and provide elasticity and mechanical stability. For example, that is magnesium activated ATPase, sodium potassium activated ATPase, alkaline phosphatase, etc. There are some carrier proteins as well. They help in the facilitated and active transport. There are quite hormone receptor proteins, for example epinephrine and etc. Now, along with proteins and the lipids, there are also present some carbohydrates that are present exterior or polar ends of phospholipids at the external surface of plasma membrane. For examples, glycoproteins and glycolipids. Now, we are moving on to models of plasma membrane. A number of models has been proposed, and the first one was the sandwich model, which was proposed by J. Danieli and H. Devson. plasma membrane is a lipoproteinous and trilaminar membrane. It is formed of outer and inner dense protein monolayer and middle transparent phospholipid layer. Now next it came to be our Robertson model. He saw no spaces for pores in the electron microgram. He hypothesized that the railroad track appearance came from the binding of osmium tetroxide of protein and the polar group of lipids. Next, we have our last model, which is known as the fluid mosaic model. Now, according to this model, it is a quasi-fluid nature of lipid, which was proposed by Singer and Nicholson. Now, the fluid nature of the cell membrane is important for the functions like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. Now, we can see the structure of fluid mosaic model over here, which is composed of all the proteins I have said before, that is the peripheral protein which are loosely attached, the carbohydrate group, the extracellular membrane. The membrane splits into two layers, that is the fridge fracture which was seen by the electron microscopy, the integral proteins, the phospholipid layer, that is the amphipathic layer. We can see that hydrophobic part and a hydrophilic part to it. Now through this model we come to the transport across the cell membrane. Now we all know there are mainly two types of transport, that is the active transport and the other one is the passive transport. Now in passive transport energy is not utilized and in active transport energy or ATP has been utilized. In passive transport, movement of substance is according to the concentration gradient, whereas in active transport it goes against the gradient or concentration. Now let us grow a detailed idea of this transportation mechanism. The transport mechanism is of two types, passive processes and the active processes. Now these passive processes is of four types: diffusion, osmosis, passive transport and facilitated diffusion. Now the active processes is of three types: active transport, endocytosis and exocytosis. And then at last the endocytosis is of two types, the pinocytosis and the phagocytosis. Let us move on to the more detailed version of this transport mechanism. To begin with the passive process, we have diffusion. Now what is diffusion? Diffusion is pulmonary gas exchange. Now this diffusion is mainly of two types, that is simple diffusion and channel mediated diffusion. Next we have is osmosis. Now what is osmosis? Osmosis is the process of moving water across a semi-permeable membrane towards ion or solute rich region. For example, we can say the plasmolysis in plant cell, etc. We have the passive transport. Now what is passive transport? Passive transport means moving of biochemicals and other atomic molecular substance across the membrane. But unlike active transport, this process does not require any chemical energy. Passive transport is dependent on permeability of the cell membrane, which in turn is dependent on the organization and characteristics of the membrane lipids and the protein. Next we have the facilitated diffusion. What is facilitated diffusion? The facilitated diffusion is also known as carrier mediated diffusion because substance transported in this manner diffuses through the membrane using some specific character to help. They are known as carrier protein. Now this carrier protein has a pore large enough to transport a specific molecule partway through. It also has a binding receptor on the inside of protein carrier to which the molecule binds itself causing a conformational change or a chemical change in the protein, that is the carrier protein, so that the pore now opens up on the opposite side. And now the molecule is released because the binding force is weak, and now the thermal motion of the attached molecule causes it to break away. So there are basically three characteristics of this carrier mediated transport system, that is specificity, saturation and the competition. We come to our active processes, and the first one we need to know is very important. It's known as the active transport. It is sometimes also known as the active uptake. It is the mediated transport of biochemicals and other atomic or molecular substances across the membranes. This process requires the expenditure of cellular energy to move the molecule against the concentration gradient. It also involves the use of protein carrier to transfer a specific substance across the membrane but against its concentration gradient. For example, reabsorption of glucose and amino acid from nephric filtered. Next, we have the vesicular transport, that is mainly of two types, that is endocytosis and exocytosis. Now, endocytosis is the intaking of materials by a cell through cell membrane and is energy dependent. Now, there are mainly two types of endocytosis, such as pinocytosis, which is the intaking of large site liquid nutrients, and phagocytosis is intaking of large sites solid particles. Next we have is exocytosis, that is the expelling or making out the materials out of the cell through cell membrane. For example, release of secretion from gland cells, release of neurotransmitter from nerve cells and etc. Now let us thus revive the function of cell membrane. So the function of cell membranes are protecting the integrity of an interior cell, providing support and maintaining the shape of the cell, help in regulating cell growth through the balance of exocytosis or endocytosis. The cell membrane also plays an important role in communication and signaling. Ions and large molecules cannot cross without assistance, as we have seen through transport. Provide anchoring sites for filaments of cytoskeleton. It also acts as a semi-permeable membrane by allowing the entry of only selected substance into the cell. Small lipid molecules such as oxygen, carbon dioxide can pass easily. Water can also freely move across the membrane. So now finally we have come to an end of cell membrane, and next what we are going to study is a detailed view of our cell wall. Now, what is cell wall? The cell wall is a non-living rigid structure, and its outer covering from the plasma membrane or cell membrane of fungi and plants, and absent in animal cells. It is tough, flexible and rigid, which provides cells with both structural support as well as a protection. It also helps in cell-to-cell interactions. Cell wall is mainly made up of cellulose, hemicellulose, pectins and proteins in plants. In fungi, the cell wall is produced by chitin, and in algae, the cell wall is made up by cellulose, galactans, mannans and minerals like calcium carbonate. Now, let us move on to the layers of cell wall. Now, the outermost layer between the adjacent plant cells is the middle lamella, which contains the pectins. The function of middle lamella is to give the stability, and they form the plasmodesmata, that is a cell-cell interaction structure. They are made up of calcium and the magnesium pectins. During cell division, this middle lamella, they are the first formed from the cell plate during the cytokinesis. Next we move on to our primary cell wall. Now this primary cell wall, it has a capable of growth. They are mainly present in young cell and they are gradually diminishes as the cell matures. They are mainly made up of cellulose, hemicellulose, and they are present in meristem, pitch and cortex. The secondary cell wall, it is the thicker cell wall. It is present inside the primary cell wall. They contain lignin for strength and density of wood. They are present in sclerenchyma, collenchyma, vessels and tracheids. Now the cell wall and middle lamella are transported by the plasmodesmata which connects the cytoplasm of the neighboring cells. This was all about a detailed information about the cell membrane or plasma membrane and the cell wall. Now in next video we are going to have a small detailed view on the prokaryotic cell wall, and there are two types of bacteria in our next video. So please stay on, and if you like our video, please do subscribe our channel and share with us what you want to improve in our next videos and what you want to learn from us. Thank you. Have a nice day.